What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we start our final unit of the first semester, Unit 6, Mendelian genetics. So we've already talked about a lot of things with DNA and RNA and protein creation, but now we're going to kind of go historically back to how we viewed genetics initially with this guy named Mendel, and he kind of set forth the path that we use today to get a lot of that genetic information. So let's dive into it a little bit here. And I want you to try to go ahead and pause this video and pull up another one. I know that's kind of inconvenient, but it's the best way to approach this right now, where you watch the video I have about the Mendelian genetics TED Talk, because it does serve as a little bit of a primer. It gets you ready for what I'm going to discuss a little bit further. So go ahead, pause this video, go to that one in another tab, watch that quick 3 to 4 minute video, and then come on back and join me so that way we can talk about the differences between genotypes, phenotypes, and some other vocab. Right, if you did leave me, welcome back. If you didn't, there was a nice awkward pause there. All right, so what we got from some of that video is overall some information about genetics and some new vocab words that we're going to have in this class. And the first word is an allele. An allele is some sort of variation of a gene. So usually like brown eyes versus blue eyes, those would be two different alleles, right? They're two different traits to a gene. And we usually represent these by a letter. So in this one, we're talking about P color here. I got capital Y's and lowercase y's. And what they did was they gave it a color as well, so that way you can see, oh, this is making yellow versus green. So the capital Y is the allele for the yellow color, and the lowercase y is the allele for green. Okay? So it's just a different variation of that gene. And we can have different alleles, but usually in this class we talk about one allele or two alleles at a time. And what I mean by that is we're talking about one kind of variation of a gene versus another variation of a gene at a time. All right, let's dive in a little bit deeper into our vocab here. So we have the genotypes. Now, genotypes are the genetic makeup of an organism, hence why it's called the genotype. It's the type of genes it has. So when we're talking about that, we're talking specifically in these Mendelian genetics things about the letters it has. What are its letters? Are they two uppercase letters? Are they two lowercase letters? Are they a mix of upper and lowercase letters? So that's the information that we're typically pulling for this kind of stuff here. So the genotype is shown here with the letter combinations. Also up here in each of these individual boxes. Do not start panicking because you see Punnett squares and you're starting to stress out because you had a bad experience with them in the past. We will get very good at them. I will work through them very slow and very patiently with everybody in person, online. I'll figure out a way to get you to get it. We're not worried about that today. All you got to know is what are the pieces around it first. So the genotype is I'm talking about the pair of letters here, or the pair of alleles. All right, going a little bit deeper. Now the phenotype, physical. Phenotype is the physical trait that the organism shows. So this could be its color, its shape, its size, something like that. So a lot of times when we're talking about genetics, we talk a lot with plants because plants are pretty simple in some of their genes and we can see some easy contrast. We will use some human examples, but they're not the most accurate ones. We don't necessarily determine eye color the way that we're going to talk about in this class because it's a lot more complicated, but it's something that you feel more familiar with than flowers or bean colors. But anyway, so traditionally we kind of look at these phenotypes as one being expressed over another, because in this case here I have the phenotype for yellow and the phenotype for green within this gene, but I'm showing the yellow, so we do have some that win over another. All right, up here at the top I have the both of the phenotypes here, or both of the traits here are for the green, so the phenotype shows as green. So we got to talk about this case scenario where Y is yellow beat green, and green lose to yellow and vice versa. But overall, you can kind of see genotype is the letters, phenotype is the color for right now. Okay, so the trait that it looks like, phenotype, and the letters that it has is its genotype. Continuing on, so talking about why one beats the other, well, some actual alleles are considered dominant. When they exist in the DNA, they are shown instead of the recessive one, or the one that's lowercase. So in this case, when we talk about dominant traits, we're always talking about capital letters. So up here, my Y up here is a capital Y, so therefore it is considered a dominant trait. When I have that paired with a lowercase, it wins, meaning that yellow is always shown over green. Within that DNA sequence for that yellow trait, that one is what's read first by mRNA during transcription over the green. So there's something that's triggering this one to be read over the green. If this one isn't there, like this example where I have two greens together, then it reads the green by default because that's all it has. Okay? So dominant capital letters is the one that's going to show. On the other side, you have recessive traits. Those are the ones that aren't shown when they're paired with the dominant, right? So in this scenario here, when I have a lowercase, right, I don't show this one because I have the dominant one that's going to win, that's going to show. Okay, up here I have two lowercases, so then it is going to show that trait that is recessive. So you can get some mixing and matching with these letters, but only get one of these things to show at a time, right? So I can have an upper and a lowercase y, but I still get the color yellow. I could have two uppercase y's and still get the color yellow. I could only get green in this scenario if I have two lowercase y's. So the recessive things, to show them, you need two recessive alleles to show it. All right. Now, talking about modeling some genetics a little bit. This one's a little more complicated to view overall. What I'm going to try to do is demonstrate this in a different way than I've done in the past for social distancing reasons. So what we're going to do is, as a class, flip a coin together to kind of see what the outcomes of things are. And the way that I'm going to do this is I'm going to have two alleles for different genes. And if I flip the coin that gives me heads, I'm going to pick the first letter in those alleles. And if I flip the coin and it gives me tails, I'm going to pick the second letter in those alleles. It makes sense as we go, so just give it a moment here. So I have my coin. Heads is going to represent the first letter. So I'm going to do parent 1 here first so you can kind of see what's going on here. We have two genotypes that we're looking at. We're looking at B for two different parents, and we're looking at T's for two different parents. And what I'm going to do is to figure out what letter is going to be given in each offspring, I'm going to flip a coin to determine that, because we don't necessarily know. It's all by random chance and probability. All right, so if I flip heads here, I'm going to end up, which I actually did, wow, called it. I'm going to pick the first letter. Okay, so the first letter in this group, that's what I'm going to pick for heads. So the trial 1, I got a heads, all right? I'm going to flip it again because I'm in trial 2 now. And this time I also got heads. So I'm going to have another capital B here representing that first letter of the group. And then a third time, by chance, I did get tails, all right? So I'm going to pick the second letter. Now some of you are saying, wait a minute, Mr. Fitz, they're the same letter. Doesn't really matter. In this case, you could actually make that observation. No matter what I flip, I'm always going to give the dominant trait. Notice how it's always these capital letters, okay? And we can do the same thing with the T's. So we go back over here. All right, we flip again. I got a tails for the first one, so I'm picking the second letter. All right, so I'm going to put a lowercase t there. I like to make my T's swoop when they're lowercase. It makes them easier to tell what they are. All right, another lowercase t over here because I got the second letter. And then the third one, some of you have already noticed the pattern. Do I have to flip the coin? No. These are two of the same, so I don't have to flip the coin. I can just leave it as is. It's a lowercase t. So in this case, this parent's genotype is always going to be a dominant trait for B and always going to be a recessive trait for T, because no matter what I pick, it's always one big letter out of this group and one small letter out of this group. Okay? Now, parent 2 is where it gets a little bit more interesting. Okay, parent 2 has both dominant and recessive traits for both of them. Okay, so I have capital and lowercase, capital and lowercase. So parent 2 is going to give us a little bit more diversity here. I'm not always going to give a capital B. Sometimes I can give a lowercase b. So I have a 50-50 chance for each one. So let's flip the coin here. Oh, we got tails. All right, tails will be the second one. So in this first offspring here, pairing these things up, the first flip for the first grouping and the first flip for the second grouping, I got one in the first and one in the second pair. So that's why I have one capital B in blue representing this parent and a lowercase b in blue representing the second parent. Okay, let's try it again. All right, I got a heads this time. So heads is the first letter. So I'm going to put a capital B there. So I got, this one has a capital and a lowercase. Now this one has two capital letters, right? Now let's go third trial, see what we get. Oh, it's spinning. That might take a minute. Come on, fall down. Oh, we got a tails. All right, so we're in the last one there with a lowercase b. So in this case, notice how we got a little bit more diversity because this parent can either give us the uppercase or the lowercase, right? We can continue doing that through multiple trials. And ideally, we should end up with a 50-50 split between these two. This one's always going to be 100% B because that's the only option I have, all right? Let's try the T's. Again, 50-50 chance on all these. So heads or tails does come into play. Ooh, it fell on the floor. And it is a tails, so I got a lowercase t. So in this one, I got two lowercase t's this time. Next one I got another tails. So another lowercase t representing that second one. Let's see if we can get a heads here by probability. We don't. We end up with 3 tails by random chance. So in this case, we actually ended up with all of these, giving us the offspring always having these 2 lowercase t's in these different trials here. If I had gotten a heads, then I could have gotten a capital T mixed in there, but by chance, that's what we ended up with. This is overall how a lot of genetics is done for us internally. We're not flipping coins per se, but we do have 2 alleles for a lot of genes, and we're either sending one or the other. Now, sometimes we have the same allele for the same gene, so we send the same thing regardless, but in those cases where we have a mixed match, that's where we can get a lot of diversity. All right, so did the coin flip from one trial impact the results of another? Well, not really, because every coin flip is a 50-50 probability. Just like every allele being passed on is a 50-50 chance on which one it is. Is it the first one or the second one? Now, in some cases, again, you have 2 capital letters. Well, if I have 2 capital letters, it's always going to be a capital letter. So that's 100% pass on. If I have 2 lowercase letters, it's always going to be a lowercase letter that's passed on. It's only those options when I have those 50-50s that I really got to pay attention. If I have a capital letter with a lowercase letter, I really got to see which one I'm going to pass on. Okay? Now, did your results from B impact any results from T? Well, we flipped the coin each time we did it, looking at each individual parent. So nothing from B actually impacted T. They were separate all the way across. So each time you flip a coin, that just determined which one of those 2 alleles, the 2 capital B's, the 2 lowercase t's, or the capital lowercase b and the capital and lowercase t, which one of those 2 in the pair was I passing on? Not if it's heads over here, it's automatically heads for this one. They are separate individuals. All right, and why did we split the letters up? Why did we kind of only give one of the B's either way? Well, that's because we only pass along one allele. Think about during meiosis when you produce gametes or sex cells. You're only passing on half your genetic code. So I might contain 2 B's in my DNA, but I can't give both B's because then that sperm cell would have too much DNA. I'm only able to give one of those, a haploid of those. So overall, we'll get a different diversity amount that's passed on to our either sperm or egg cells when we're producing them through meiosis. All right, so more vocabulary that we kind of saw was the law of segregation. Now this one, segregate means to separate. So in this one, this was the coin flipping. I have 2 alleles for each gene, all right? 2 capital Y's here, 2 lowercase y's here. When I make sperm or egg cells, those are split apart. So I have a capital Y that goes here and a capital Y that goes here because they're the same thing, right? And a lowercase y and a lowercase y. If I have it where it's a capital Y and a lowercase y, one sperm cell that's created will have a capital Y, one sperm cell will have a lowercase, so you have 50-50 between them. All right, and this is all because of meiosis. Remember, diploid cells have that full set of DNA. Sometimes it's referred to as a 2N set. So you have 2 alleles per trait, right? I got 2 eye color alleles, I have 2 hair color alleles, all that stuff inside me now. But when I produce sperm cells or females produce egg cells, that DNA number is cut in half, so they're only getting one of those traits. They don't know which one it is, right? One trait for eye color instead of both. That way, there's a half set of DNA that can now be used during fertilization when sperm meets egg to produce a new offspring, okay? So haploid cells have 1 allele per trait. That's why during meiosis, we cut this number in half, and we do it randomly through this law of segregation. Each trait, each genotype has to be separated in 2. And this is why in Punnett squares, you put the 2 on the sides, right? You're separating them through the law of segregation. And we'll explore that a little bit more when we get into Punnett squares. All right, the law of independent assortment is a little bit more interesting, but the base principle is no matter what happens with one gene, it doesn't impact the other gene. So if I pass along Y, it doesn't impact how I pass along R. If I get a capital Y in one, I could have a capital R in one. It doesn't really matter because Y's pass on, right? Y's coin flip does not impact R's coin flip. So I can get different variations here, different variety. Just like in the experiment that we did, a little example, the letter I got for B didn't tell me what T was going to be. I had to flip the coin individually to see that. So that's that independent assortment idea. Now, putting it all together. So thinking about, did B change T when I flip a coin? Does it impact each other? No, because each trait is passed on individually. That is that law of independent assortment, that it has to look at each genotype alone. You can't determine, well, capital Y went over here, so it's got to be capital R over here. Lowercase b went over here, so it has to be lowercase t over here. That's not how it works. You look at each trait individually, you look at each genotype individually. So each time that you're trying to figure out the probability of passing something on, you're going to have to do a Punnett square for each trait and then figure out what the probabilities for each of those are and put those together. That's some complex math that no one wants to do, trust me. Where do we get our traits? Well, that one's pretty easy. We get our alleles from our parents. We get one allele from each parent to make us having two alleles for each gene. So we get a genotype based on what alleles were passed on from our parents. Each one gave a single one, they come together, and that makes our genotype there. And then what do the alleles hold information for at a molecular level? So thinking back to the last unit, they hold the information to make proteins. Remember, genes hold the information to make proteins, and through the central dogma, those genes are made of DNA, they transcribe into RNA. Those RNAs are then translated into proteins, and that's how we actually get all of our traits overall. I know this was some heavy stuff. Please rewatch the video as necessary. We're going to get into more vocabulary tomorrow as well in the next couple of videos when we start talking about Punnett squares too. Don't panic, stay with it, and I know you can do it. That's all I have for today, and have a great day.